Everyone, hello everyone. Thank you for joining us again this month for our monthly cell modeling seminar. This month we'll be listening to Dr. Stephen Larson. Dr. Larson is the co-founder of OpenWorm, which was a collaborative project to simulate all cells in C. elegans. He's also the CEO of Metacell, which creates software for exploring neuroscience. He has experience both in collaborative model development and in simulations of a whole organism. And we feel that these are two issues our community needs to address as we scale up our models. So let's listen to Dr. Larson.
All right. Well, thanks very much for that kind introduction. I'm going to be talking to you today about OpenWorm, a project that I've been grateful to be involved in for the better part of 7 years now. But it's really the work that I'm going to be talking to you about today is also the product of a pretty significant community. At this point, there's probably been at least 100 people who've contributed to the project over the last 7 years. So while I've been in a position to help shape that energy in a direction, really I'm going to be showing you a lot of work that is not my own, but I'll call out some of the pieces that are mine as we've been part of this. So let me figure out real quick how to share my screen. Okay. And let's see, folks seeing a slide cover? Yes? Okay. So, so I'll be talking to you today about OpenWorm. It's open science for C. elegans modeling. And so we'll go into the worm, as you see here, and figure out what's going on with this. Right off the bat, I do want to give a shout out to some of the collaborators and supporters that have been working with us in particular. So we've got active collaborations with researchers around the world, ranging from such geographically diverse places as Novosibirsk in Russia to London, several facilities in London, to folks in Vienna, folks back in San Francisco, folks at ASU. And in addition to those, we've been grateful to receive some grants and supports from some of the leading tech industry companies. So we've gotten research grants from AWS. We've had several students now come through Google Summer of Code and work with us over about, I think, four or five summers now, have worked with us. So what I'm saying is really the product of a whole community of folks. So the idea for OpenWorm begins where I think a lot of reproducible modeling begins as well, which is this desire to bring an engineering mindset or mentality to something that we have not as human beings actually built, which is biology. And we take a lot of inspiration from this approach that Richard Feynman had left on the final board when he passed away, which was this idea: what I cannot create, I do not understand. And this is our major challenge because, in fact, we did not create living organisms, and therefore we have this challenge of being able to understand them. So modeling steps into the breach to give us the ability to attempt to rebuild, to recreate, to reverse engineer what is there and bring it back together into a synthetic whole. So our project in OpenWorm is to build the world's first digital organism, multicellular organism now, as we're sort of the space for single-celled organisms is heating up. And what that really means is not just a single digital organism or a single instantiation, but actually what the project has evolved into is a framework for a comprehensive computational model of a particular animal, this very well-studied nematode worm, this microscopic worm, in close collaboration with the scientific community. So I'd like to pose the question today in the following way, which I think also generalizes to other kind of biological modeling, which is if we had the ultimate microscope, what would it show us? What would it be able to show us about how an organism works? And so when I'm thinking about the ultimate microscope, I basically mean, if we had the ability to see all the dynamics that happen inside a biological organism in great detail. If we could change our focus and see how all the neurons are adjusting their membrane potential, how calcium levels are changing within the neurons, but even down at the level of muscles, how calcium is moving in muscles, all the states of the proteins of the organism. We could observe different kinds of molecular activity happening throughout the cell and throughout the organism. What would we be able to see? So it's from this mindset of, and this perspective, that there is an unseen reality of biological organisms that we approach this project. And so in the early days of the project, when we were longer on concept and shorter on results, the results came later, we put together images like the ones that you see here for a Kickstarter video that we launched several years ago now, but that it tried to capture this sense of the ultimate microscope and what we might be able to see in this sense. And so to really do that for a multicellular organism would then require us to be able to, be able to see sort of transparently through the living organism as it moves. Thankfully, our worm, C. elegans, is already transparent, so that helps. But we'd be able to analyze how activity would flow through it. We'd be able to see how its muscles bend, and we'd be able to see these different activation patterns in general, and then hopefully use that as a foundation to be able to extract more theoretical understandings of how these organisms work. And so that's really the second piece, which is to be able to get a sense of what that microscope would show us. We'd perhaps be looking at an organized mess of activity. So we would want to take the next step and to be able to extract a deeper understanding, a theoretical understanding, a mathematical understanding of how all those dynamics work together. And there's many different forms of this question that we could ask. We could ask, what's a principled mathematical way to understand how digestion works, or principled understanding for how reproduction works. But we got interested in particular in the nervous system as a focal point for this project. The C. elegans has 302 neurons. It is about as long as a hair on your head is wide. It is a pretty easy to work with animal in the lab for many reasons. So we thought perhaps that the beginnings of a new kind of understanding of information processing of the nervous system is possible in this animal. And of course, there are many labs around the world that study this purely biologically, and a few that do computational work in this space. But there's not a lot total. And so we felt that perhaps we could help make a contribution in that space. And we're inspired by software in the engineering space that is able to give engineers a very deep understanding of large-scale devices. So on the top here, we've got cutaways through the Boeing 777. Something like 2 million parts went into building this aircraft, and a large part of the design and testing was done in software where all of the pieces and components could be subjected to stress and strain in advance of building it. And it saved Boeing a lot of money on that in being able to run those tests virtually before having to run them physically. And in the bottom, a similar idea to sort of zooming down into a motor of a plane. So inspired by the fact that software exists that is able to give us these really detailed views of breakdowns of the cutaways of millions and millions of parts, we figured, biology should be able to do a similar thing. And I think to this audience, this is not an unfamiliar concept. And so we take inspiration again, as I was saying before. So here's the Jonathan Carr slide. So we take inspiration from work that has, gone outside of the scope of the field of modeling per se, or the field of biological modeling per se, and in fact has broken out into the field of biology. So one of the things we were really excited about in this particular paper was the fact that models were getting cited in non-modeling journals. Of course, this has happened a lot, but for an audience that hasn't been familiar with this idea, the idea that we can reverse engineer even very simple organisms, and be able to make reasonable predictions about them, even if it's coarse-grained and even if it comes with some uncertainty. Holistic modeling in biology is something that has been gaining traction, and we thought perhaps we could go from this single-celled example to a multicellular example. And so as a product and result of the work that we did on the Kickstarter, we were able to begin to bring some features or components of C. elegans modeled first physically, also some nervous system simulations to an environment that we called WormSim. And in this case, we were able to demonstrate a simulation where we are just putting a very simple sinusoidal bend through the worm, but doing it by articulating 95 body wall muscles of the cell and having the result of the contractions lead to the sinusoidal bending through a detailed physics simulation, which I'll tell you more about in a little bit. But this is still pretty early days because a lot of this was made more from an educational perspective than from doing hardcore modeling. But at least, at this point, we're starting to, and around this is like 2015, we're starting to be able to expose some things to the web and get people excited about our work here. So we've done several sets of projects over the last 7 years, of which I don't really have time to go into all of them, but I will give you a little bit of a grab bag of some of them, and then go into a bit more of a detailed approach in a few. So one of the things we first started out with in this project was something called the worm browser. And here we were really not even focused on simulation yet, and not really focused on any kind of dynamic modeling. We were just focused on taking a really good three-dimensional data set, three-dimensional atlas that had been produced, and making it easy to use. And sometimes in this space of reproducible modeling or open source sharing, sometimes just a product that people find easy to use makes things a lot better for a community and begins to form a focal point for a community. So we crossed over what used to be the Google's body browser, and then they discontinued it, and we reused the open source software for that, plugged a worm into it, and we actually got hundreds of worm scientists around the world checking this thing out, using it to figure out their three-dimensional movies that they started taking through the head of the worm to identify neurons in there. And so this was first major effort that came out of this. Then going beyond that of course was a rigid model of the worm's body, which featured every cell that was known in that atlas. A project called Cybernetic, which was chiefly led by the folks in Novosibirsk, Siberia, hence the name, focused on integrating biophysical knowledge into a fluid mechanics model. Here we took a subset of the cells, mainly just focusing on the body wall muscles and the cuticle, which is the outside skin of the worm, which tends to be under pressure, and focused on the mechanics of how muscles bending can lead to changing the shape of the worm on the whole. And so that meant that we needed to know things about how tough the cuticle was and how bendy it was, and the force that each muscle actually was pulling, put something in a scale that was reasonable. And this also interfaced with either fluid or gel environments. And so this now gave us a whole nother perspective, use a whole different kind of math in order to make this work, all with this goal of understanding the physical situation that the worm finds itself in. A whole nother slice of work that we've done in this space is something called PyOpenWorm. It's a Python library for accessing data, primarily at this point about the nervous system, but the idea being that we needed to decouple the modeling effort from the pulling data together that we were going to build the model from, with the idea being that if we essentially had a database of information, we could generate an arbitrary number of models, but the information that we were basing it on, such as the name of cells and different things about the connectivity patterns of cells and whatnot, should live in its own space. And then different kinds of models can be generated from that, but there's kind of a ground truth. So PyOpenWorm is this open Python library that we pulled together that basically wraps a database, but makes a consistent interface to all the things you need to know, primarily at this point about the neurons of the C. elegans. ChannelWorm was a big effort to pull together information about ion channels, which for the kind of modeling that we were doing of the nervous system, primarily conductance-based or sometimes called Hodgkin-Huxley-based modeling, is a pretty important dynamical component of this. There's not a lot of coverage in C. elegans of the dynamics of individual ion channels. So we were doing some work there to pull what we could out of published journal articles, as well as trying to make the case to generate some new data in this space as well. And so here we kind of built an annotation interface for pulling those papers in, annotating the IV plots that come from papers about ion channels, such that we could extract key IV curves and use them to constrain some parameter sweeps to figure out some reasonable models for ion channels. And then movement analysis on a whole nother level of data. We're working here with open source tools that help to quantify behavior. So if you imagine a camera that's looking down at the worms as they're crawling around on dishes, there's a bunch of tools in here to do analysis of how fast they're moving, how often they turn around, how often they move forward or backwards. Definitely things that you need to constrain body model of the worm so that we know that we're reaching movement patterns that are anything like the real animal. So as an example of some things that we might pull out of these data and some of the work that we have been doing in terms of our simulations, so we compiled, for example, the velocity reported in several articles of the worm crawling over surface with a gel. We looked at the wavelength and the frequency. These kinds of coarse metrics of movement were something that we needed to be able to know what's there printed in the literature. And happily, we're able to report in papers that came out this fall, sort of hitting within reasonable ranges. And of course, any simulation can hit within a reasonable range of one parameter or two, but this is a selection of like 3 out of several dozen that we were matching with pretty good fidelity all at the same time with a model that's constrained. And this is the cybernetic model I was talking about, which is the fluid dynamics environment. So kind of demonstrating that we're at least beginning to get some reasonable matches, at least at the level of being able to demonstrate that we can generate something that looks like crawling. At this point, still separated from a nervous system, but beginning to go down that road in what I'll show you next. Okay, so I mentioned the worm browser. This is what that looks like. Basically an interface to sort of zoom through the cells of the worm, and you can rotate it and pan and zoom, and you can check it out at that URL. I mentioned PyOpenWorm, a little bit more about that. It uses a graph database as a backend. And basically a lot of this work is taking spreadsheets, turning them into a form where they can all get integrated together so that you have apples to apples comparisons between data. It's very helpful in this particular organism that the cells all have a naming convention, and all the cells are often conserved. So you can basically go from worm to worm and look at specific cells and know which one you're talking about. A little harder in a human or other animals. And so reconciling the data into a single graph has been very helpful for us to be able to use that as a foundation for modeling. And so we've integrated these different data. Much of it comes out of wormatlas.org. We've gotten data reported from collaborator labs in some of the articles that you see here. And what do I want to say about this as well? So I think the range here that you're seeing coming from specifically in the neurons that we've integrated into PyOpenWorm incorporate things as diverse as the connectivity patterns, as well as the receptors that are held there. Innexins are terms for gap junctions, physical connections between cells. And so again, they're all kind of organized in a way where you could build different families of models if you'd like out of this kind of data. So you can choose to use this data or not use this data as you like on your model building side. So we have the ability then to programmatically get access to this data so that our researchers can use it either to ask questions for their own papers or to generate models as they like. And that's what we do in a platform that we call, for our purposes, something called C302, which I'll say a little bit more about in a moment. But a lot of this stuff is written up in papers that came out in the fall in the transactions of the philosophical transactions of the Royal Society B, in a special issue that we were involved with getting out. So you can read more about that. You can read more about PyOpenWorm in that particular article. I mentioned a little bit about ChannelWorm as well. Don't need to go into too much detail, but I mentioned the annotation interface that allows us to identify the details of individual ion channels. And so there's some code there. You can check all this out online on GitHub as well. Some other community efforts, if I take a step back, that have also been involved in this. So we put a lot of focus on our scientific roadmap on a particular perspective on modeling the C. elegans, but we've also had sort of sister projects spin out of the project, including DevoWorm, which is a project that's focused on the developmental lineage and the development of the C. elegans. It turns out that every cell division from a single fertilized egg has been mapped out also for the worm. And so folks have been experimenting with other classes of models, like cellular automata type models, to see if we can find ways to build a generative model of development. There's several DOIs listed here that you can find out a little bit more about those things as well, in terms of morphogenesis and embryogenesis of the worm, and some modeling efforts there. And ultimately, we'd love to have a simulated worm that starts from an egg and grows up into an adult, and then goes on behaving with an active nervous system and an active muscle system. That's kind of a longer road into the future. And then also for educational purposes, so OpenWorm is actually a foundation. It's a 501c3. And in addition to sponsoring research and being a vehicle for doing the kind of research I've shown you so far, we also have a strong educational mission, and as a way of promoting not only open source software, but also open hardware. We've have a project to do some open hardware robotics, of which you're seeing some examples here. So one of our repos on GitHub has 3D printable models where you can print out little segments for a worm that rolls around on the ground. Of course, it's very different, biologically speaking and functionally, than what we have in simulation, but it's a pretty cool thing, and we find folks get pretty excited about checking it out just to get inspired about this kind of modeling. So that's pretty cool as well. Okay, so I've kind of talked a lot about things from the perspective of code repositories. Now I just kind of want to circle through a little bit more scientifically, like a line of research that we've been doing that integrates these different code repositories together. And I sort of suggested it at the front of my talk. Basically, we want to know, if we're simulating a small nervous system, can we understand information processing principles about how it works? That's really what drives a large amount of the efforts that we've been working on here. So I've kind of touched on the different components of what goes into the main model that we've been conceiving of, but this picture by Untitled, published way back in 2011, I think does a really good job of summarizing what we're looking at. So this particular article made the case that a lot of computational models had historically been operating only in the realm of simulating neural circuits without really integrating behavior and biomechanics in the loop. And we were pretty inspired by that when we first started, having built many of those kind of disembodied neural circuit models, thinking that, if you could ever reincorporate biomechanics in a reasonable way and get down to some pretty constrained physics, this would be a great organism to do that in. And so, in fact, as I've sort of been alluding to, we've combined a nervous system circuit model with a biomechanical model that includes muscles, body, and an environment where the loop is essentially closed to enable those systems to talk to each other all together in a simulated environment. And we feel that this is, although a rather complex set of things to capture, what we feel is that, and this article by Untitled articulates that if you leave any of those components out, you are missing what could be some really critical dynamics for really understanding what the neurons are doing at any moment in time. So we want to make sure then that what this allows us to do is to take very different kinds of data and use them all as constraints for this model. So data as diverse as movement analysis data, like what I showed you in terms of the overall speed or frequency or velocity, down to specific gaits, down to the activity of muscles, the calcium activity of muscles, the calcium activity of neurons, and then even things that we could reasonably infer about membrane potentials at given moments in time, or at least the bounds on those things, all allow constraints for the system. And for anything as complicated as a whole organism, the more data that you can bring to bear and the more constraints that you can put on it, the better, right? In general. And even though it's still probably under constraint, even with all those data, again, we're looking for a way where we can get it as constrained as possible, and then potentially put the challenge back to the community of, what are the data sets that could still be collected that are the most informative, that move us in the most informative direction for behavior. So anyway, this is the scope of simulation. So let me talk a little bit more about what goes into that. All right, so I've sort of mentioned some of these terms as we went. So the part of the system that's capturing the nervous system as well as the electrical dynamics of the muscles is called C302, and the part that does the biomechanics is called cybernetic. Those are the chief simulation engines of OpenWorm. So one of the things we're able to do is to explore some hypotheses with two different kinds of motor neurons, the neurons that are synapsing directly onto the muscles. So this is basically the neuron that tells the muscles to flex. So if we imagine building up a layer cake with muscles at the bottom, we're representing the muscles here as these individual red ovals. They run all the way from the head of the worm down to the tail. They run in these 4 quadrants radially along the body of the worm. So you imagine that each one of those gets about 6 to 8 synapses from different types of motor neurons that have been identified. Those are listed here, so you can imagine connections coming out from these motor neurons. And so we began just sort of saying, okay, worm is basically crawling like a sinusoid. We spent quite a while engineering the muscles to contract in such a way as to produce the sinusoid of the body. Also, basically, it's sinusoidal input. But then we took the next question and said, well, okay, given what we know about the connectivity patterns between all the motor neurons and the muscles, what is the pattern of activity that the motor neurons must be doing in order to stimulate the muscles to have the sinusoidal pattern, right? And again, you imagine, well, it's some sort of transformation on a sinusoid, but given that the connection pattern is not completely homogeneous, it becomes a little bit more interesting to say, well, what is that pattern? So we began with a modeling scenario where we knew that we'd be doing things that were not going to fully reproduce reality and not fully reproduce biology, but we wanted to at least be able to get this entire system of body plus nervous system into a place of tuning parameters that we did not know, so that we could get the kind of output behavior that we were interested in, or at least get close to. So again, we picked this subset of cells. This is a different way to look at it. So up here is the worm. This is in green are the muscles going from head to tail. This is what it looks like in a more anatomical form. This is what it looks like in the 3D Atlas, the worm browser. And these arrows here are just showing that there is typically thought to be a sort of proprioceptive loop with the environment in this space, so that the worm actually knows how bent it is at different places as a result of some special stretch receptors and neurons. And down below is a schematic diagram showing on the top the layer of muscles and the bottom layer of muscles, so dorsal muscles and ventral muscles, the DM and the VMs. And then inside of there are the motor neurons. The DBs and the DDs are the dorsal B-type and the dorsal D-type motor neurons, and the ventral B-types and the ventral D-types. And these are connected together. And then we picked one interneuron that's known to be really highly connected, one of the most connected neurons in the entire nervous system. And we put its connectivity into the system. And then those V-type and B-type, you think of them as those motor neurons also going down the body of the worm as well. So what did we do with that? And this, by the way, is in a paper that we got at NIPS in 2017 in a workshop that we did there, which is pretty cool. So what are we doing in this process? So we are starting a process. We had started a process at this time of doing some automated parameter optimization with things that we didn't know, certain conductances or basically the weights between the neurons are not fully known. We know that a neuron A is connected to neuron B, but we don't necessarily know how strong that connection is. And in fact, it's very likely that it's variable from individual to individual. So it's really not fully knowable on the whole for every single worm. So given that there's variability, we turn to a combination of automated parameter optimization as well as some hand tuning to search for free parameters. And after many iterations, we came up with a pattern of activity that would allow us to begin to see the worm crawling. So what we're looking at here on the left is the membrane potential simulated of 39 neurons. That includes the motor neurons that I just mentioned, as well as the left and right of the inner neuron. And on the right are the simulated calcium activity. So we do have calcium as part of this, simulated calcium activity of the 95 muscle cells. And so the way to read this raster is we've got every row on the y-axis from top to bottom is an individual cell. And we've got time going along the x-axis. The color code is showing the intensity, so either highly activated in warm colors or not very activated in cold colors. And so the striping effect that you're seeing happening here is showing you essentially as we're running, that's the other thing, is that the striping is showing you as a result of organizing the cells by order from head to tail, depending on which quadrant we're on. So the pattern repeats because we're choosing to show all of the motor neurons of one type all together in order as they go from the head to the tail of the worm. And so this shows you that things like a sinusoidal movement is a result of, in this particular instantiation, of activity kind of moving from head to tail in sequence in the right ways. So it was pretty cool when we got the muscles to do that just on their own by feeding in a sinusoid. It was even cooler when we were able to generate this pattern of motor activity on the neuron side and have that lead to the muscle activity. And so what does that look like in practice? So when we feed this pattern of activity into our simulation, we have this. And so what I had showed you at the beginning of the talk was movies that we had made that were concept art. And what I'm showing you now are the three-dimensional renderings of a physics engine. So it's a very different story now, which is quite cool. On the left-hand side here, we have, this is real data. So this upper left is a real worm with a real camera. It's got an overlay on there that comes from computer vision, which is why it looks kind of a little somewhat synthetic, but it's a real worm. In the upper right-hand corner, we have the sinusoidally injected, like only the muscles are being instructed to make a sinusoid with a simple sine generator, basically. But we have a 3D view on it, so we're looking at it from the side, and this is our cybernetic simulator. And then on the bottom, we have the same simulator. This looks like a little different because it's different colors, but the same simulator engine, but now it's crawling around. Of course, it's in a bit of a constrained space, but it's crawling around, generating that sinusoid as a result of the motor neurons giving us that sinusoid. And again, sort of reproducing some pretty good forward crawling behavior right there. Believe it or not, there's, a lot of things that you don't see here about things that didn't work, but just to appreciate, things that didn't work. So in early days of cybernetic, the fact that it's a shell that has pressure, hydrostatic pressure, pushing out on it to create its rigidity, which is true of the actual worm, that didn't always work. Sometimes there were things like the innards of the worm would leak out and the worm would deflate like a souffle and therefore not be rigid anymore and therefore not be able to move forward. So that is a possible failure state of this simulation, which we over time fixed. And then even things like being able to crawl forward There were many iterations of this model that just sat there and just wiggled. So the ability to actually carve a path through the substrate that you're seeing here requires us to have modeled the substrate pretty well, requires us to model the interaction between the skin of the worm and the substrate in a reasonable way, so there's enough sort of pseudo friction in the system to make it work. So while you may look at this and be like, okay, well, cool, well, I expected it was going to work, actually there's like dozens and dozens of things that had to be accounted for in order to produce, you know, this pretty satisfying video. So that was pretty cool. We reported this as well. We just got this out also in the fall in that same special issue of Philosophical Transactions. This one's an article by Gleason. So feel free to check it out. Some good stuff in there as well. Okay, and we've actually bundled all of this that I've just showed you, and even the ability to generate a movie like that into a Docker container, which is on our main GitHub page at OpenWorm, github.com/OpenWorm/OpenWorm. If you want a little bit of a intro to that before you go try it out, and I think there's always a conversation going on about how to iterate it. You can go to this URL, which will drop you into our Slack channel. Right now, the community is really centered around our Slack organization that's there. If you don't get, if nobody says hi to you, or if you don't understand what's going on, I encourage you to nag everybody until somebody answers you, including me. You can find me in there, and you can just direct message me mercilessly if you like. These days, we're reworking how we're doing our community stuff, so you might find yourself in need of doing some nagging, but rest assured we are active and things are happening. So just jump in fearlessly and begin asking questions. Also, we're currently looking for projects for Google Summer of Code 2019. So if you want to get involved that way, we definitely have some ideas cropping up that you could apply for if you're a student and join us this summer. And otherwise, you can check us out on Twitter, Facebook, our YouTube channel. I think we need to refresh some stuff. We haven't posted there in a little while, but there's always stuff happening on GitHub. And yeah, so I think that's the main thing that I wanted to talk about. We have some additional directions for the science where we want to go one step beyond the motor neurons. We want to really understand what's going on at the level of interneurons, and I'd love to talk to you more about that as we go, and so we can really close the loop with the full nervous system. We'd like to do some work with more interesting behaviors than just crawling forward. We'd like to have it crawl forward, have a stimulus like a tap of the dish, and then begin crawling backwards and be able to model that entire process, and then be able to mine that again for an understanding of what's going on in the nervous system to produce that in the interneurons. And that's kind of the next directions that we're headed towards. And lastly, just for contributor purposes, again, I sort of mentioned there's like been about 100 people who contributed to the project, and the only way that I can cite them is through a scrolling movie credits like animated GIF, which is what this is right here. So all the people that have been involved in the project over time, as much as we can, have been put on a big list. We also actually have a whole nother list of folks that have contributed to us in sort of resource support way, but these are all the rest of the folks that have been involved with us. So I will stop there and open up the floor for any questions that you might have. Thanks very much for the opportunity to talk.
Thank you, Dr. Larson, so much for your presentation. I guess to start, anyone have any questions on the presentation?
I may ask something. Some things that you're showing are very similar to what people were doing maybe a decade ago, last decade or two, with actually machines. They were trying to make artificial life from machines, computers. You are, however, are trying to do this on a trying to imitate a living organism, where they were trying to imitate how would be the evolution of a living organism, but using it from mechanical building blocks. So you kind of are taking it in different direction. Can you comment on this?
Sure, I think so. You may be picking up on like an artificial life flavor to some extent of the work that we're doing. anytime that you're reincorporating a body model or a physical model, it does harken back to work where there was only a physical model of an animal, an organism that was used here. And so there's definitely a component of that. There's folks who work in this space, in particular, I think of Randall Beer, who collaborates with Eduardo Izquierdo. And Randall Beer is someone who is very much published in that space of artificial life. And so we're definitely inspired by that as well. I guess, here we are doubling down on the fact that we know a lot of the specific biophysics of this particular animal, and we're really trying to reproduce a specific one. We even are doing the specific gender of the animal at a specific life stage, with specific length and specific neurons and specific cells. And so we're trying to just take that idea and make it, much more specific in that space. And I will say that, we are not the only people working on models like this, and there's a lot of great modeling happening in this space. If anything, what distinguishes us is we're trying to pull together a lot of different kinds of models and make them all interact in a single place and expose all that, into the open source. So we definitely reuse methods that have been out in the space for quite a while. We're also reusing a lot of methodology from computational neuroscience, which is also 10, 15 years old. So we don't really view what we're doing as breaking completely new ground on all fronts, but more trying to piece together the best of what's already happened. Even data that's been generated 5 years ago is exciting to us if we can pull it together and aggregate in this space. In that way, as a foundation, as opposed to maybe like a research university, we have slightly different incentives. We're interested in this holistic perspective more than like the latest, greatest. So I hope that answers your question at least a little bit.
So hi, can you hear me?
Yes.
Yeah, so I got one question. I've been following you right from day one. I guess the one question I have is, how do you manage the distributed nature of the project? You've got people scattered over. I'm just interested in the logistics and the mechanics. I notice you use Slack. So has that been important to you?
Yes, it has been, and we've got it's both been better at certain times and more challenging at other times. We've innovated a lot on the model of essentially distributed collaboration on science. We have several standing committees in the organization that work on these problems. In particular, we've got a community committee. So there's a set of tools that we like to use in concert with each other to facilitate this. So defining documents in Google Docs. We use Trello quite a lot, the Kanban system for collaboratively arriving at tasks. A heavy reliance on GitHub, of course, and GitHub issues. We integrate a lot of things into Slack so that when something happens on one of these other places, like when something happens in GitHub, somebody commits something, there's a notification that goes into the right channel in Slack, and then everybody knows that it's there. We do our best to have regular meetings on a weekly or monthly cadence so that there's always a reason to confront what's happened in the community on a regular basis. And we're trying to do more and more with like automated reminders that a meeting is coming up, so that even though we're not physically together and we can't tap each other on the shoulder, that these things happen. However, I will say that it is a challenging thing to manage and coordinate. So sometimes it works better than others. But yeah, right now I'd say Slack is probably the hive mind. It is the place where we all kind of collectively get together and work. And then through like hangout meetings, to augment that, we coordinate.
All right, thanks.
If I can follow up with that question. And I know during your presentation you mentioned that C302 worked on the neural network aspect of the model, and Kinetics worked on biomechanics. So it sounds like that these two projects were both delegated to different groups to work on, but then were integrated at the end when sort of biomechanics informed how the neural networks were parameterized. Practically, how did you manage to distribute both of those projects and then integrate them back in at the end?
Well, distributing them was easy because there were different teams that wanted to do those two different things. So the initial division of those two projects was very natural. The combination required more work. In fact, it was a long-standing gap in the project that these two did not, quite interact. So the integration had been more difficult. But we finally achieved it in the last couple years, which we're very pleased with. There's still more to do. So we've got nervous system to muscles working, and we're working now on sensory information coming from the physical model and going into sensory neurons. But we have a path to that. And at the end of the day, technologically speaking, the fact that both systems have the ability to interface with Python was actually really important. So Python is the glue language that it is. Everybody's nodding. Yes, Python is a good glue language. Open source technologies in general being really important because it allows for lots of creativity in this space. Whereas, if this was all written, I guess if everything was written in MATLAB, that would be okay. But inevitably, that if somebody wants to write something in some other language of their choosing. So yeah, and I guess beyond that, it's just we hosted a lot of sessions with these teams in the same virtual room talking about how we were going to do it, and through GitHub pull requests, successively. And I think we at some point had three different competing approaches to reintegrating those two code bases, and we basically let each team kind of compete. So we said, okay, nervous system team, you build an integration to the biomechanical side. And we said, biomechanical team, you build an integration to the nervous system side. And then everybody kind of did that for a while and was all committing code. And then kind of the third way ended up being the compromise solution between the ways that they wanted to do the integration. So in the end, it was still kind of the distributed teams doing it, but intersecting finally when they stared at each other's code and they were like, oh, no, it'd be better if you do it this way, or it'd be better if you do it that way. And so, yeah, the integration part is typically the problem of the distributed project, is how do you reintegrate? And the more that you need to integrate, the more your leaders and your leadership need to be able to convene sessions like the ones that I'm talking about, the more time it takes. And so that tends to be the bottleneck then of reintegrating the pieces.
Interesting.
Looks like we have a question from Samuel, if you want to jump in.
Yeah, thank you. So I was wondering, how did you do all the data integration? Because you have all these different sources. How do you actually get that into, your database? How do you verify that indeed that these are the correct values and the uncertainties that go along with them?
Yes. So we have an ideal case of where this happens, and then there's a lot of unideal examples of it. So I can't say that we've absolutely done that as well as we could. But what we've done in PyOpenWorm that I showed earlier is an attempt to do that as well as we could. And so what we did there was we took great care to have copies of the raw data out in the open source, right? So that's the original spreadsheets. And then the CSVs from there as well, also on GitHub, so that they're fully there. So within PyOpenWorm, because data sizes are small, we actually have the raw data all inside the repo. And then we also have the code that does the integration. It's Python code. And so when you build that project, it actually, for everybody who builds it, you're actually rerunning the integration on your own local system into this graph database. And then on top of that, we took a lot of time to build a lot of unit tests that serve the role of kind of like data checker unit tests. So for example, it'll say do I in fact have 302 neurons in my database? And if not, then that means somebody screwed something up in that integrator script where we left out a cell and where is it? Or just other sort of basic sanity checks. It's something I hadn't seen as much before, to do data validations and build them all into the same repo. And so that was kind of the primary strategy for us to be able to do data integration in this way, that everybody could see how it worked, all the steps. And so we have a documentation page that you can actually click on all these things, and you can see for all those data sets, what's the original, what's the code that processes it, and and the citation of the paper, all that stuff is at least tracked for that part. And then for some of the other pieces, like on the biomechanics side, we don't do it as well. But this place, at least, is a place where we've done a reasonable job to start with.
Thank you.
Sure.
I did have a question as well. So I understand that the holistic approach is what really drives the project. And I was wondering when you want to integrate new data, how do you, so if all the pieces work together and click well, great. However, oftentimes, having dealt with a similar issue, this new experimental result will, in order to accommodate it, you will need to tweak a few parameters, and it may be in contradiction with other experimental results. How do you go about doing that? Do you use some kind of optimization framework, and if so, which one?
Yeah, I wish I could say that there is a silver bullet and a single way to answer your question. Unfortunately, there is not. So I will tell you what our philosophical approach is to the project that helps us with that problem that then we end up solving case by case. As much as possible, we've tried to keep a pretty close correspondence between code that we have in the system and biological reality. So so for example, we try to, as much as possible, represent each neuron as a thing. And all the aspects of that neuron, as much as we can pull them together, are all kind of kept together like objects in code. So what that means is when results come out and they say something about neurons or some aspect of neurons, we will try to bring that data into register with the actual biological entity that the data speak to. If we have data that's speaking about the movement of the worm, we keep all that. We keep the way that the representation of the worm's body all together in the same place. So because we have, this is kind of the benefit of having a very detailed model of this organism, is that we're representing the geometry of the body of the worm. So some data may come out and only represent the speed. Other data may come out and include the width of the worm at different points in time that wouldn't be present if all you were doing was modeling the worm as, say, a single line or one like a two-dimensional thing, not a fully three-dimensional thing. So because we've taken these pains to have these representations that map to biological entities as much as possible, it gives us more flexibility when some new data set comes out from an experimental method that we hadn't even thought about, right? And it gives us a place to start getting in there and working on it. That's one answer. Another answer is that we have built several of these pieces on top of reusable standard frameworks. So what we're doing in C302 wraps about another 15, 20 years of work on reproducible nervous system models that goes under the name NeuroML, which has been part of the SBML families that I think folks have, some folks have considered here. So doing that means that when, we want to add, calcium dynamics in, it's pretty straightforward to get a calcium model and incorporate it into what's there right now. As we go down into like channel rhodopsin experiments where we're stimulating cells with light, there's some existing channel models for that. So at least in the nervous system, we do have a little bit of a plug and play kind of environment where we can define the model declaratively. And that helps for that kind of a space. But other places where there hasn't been as much effort, like in the building a physical model of the worm's body, it's not standardized yet because there's not really any other version of it than what we've got. So we do have to, so if we learn something about the Young's modulus, which is a term that refers to the bendiness of a physical thing. So there's some data comes out which took the worm and bent it and tried to measure how elastic or bendy it is. We may have to kind of find a way to make sure that our model is, we may have to basically build a new test to prove to us what that metric is in space. So we may need to literally have a new simulation which takes the worm and bends it, and then inside of our model we're measuring what the physics say about how elastic it is, basically reproducing the experimental method that was used to gather the data in the first place. And then we may have to say, okay, wait a second, so our model is actually 5 times more bendy than what the real worm is. What does that mean about the other constraints that are in the system? Is the skin too elastic? Is it too thick? Is the thing too heavy? And this is what we did, many, many times. So the only, again, I've now given you like a few examples on the nervous system side and on the body side. There's not a lot of correspondence between that. The only thing that really corresponds is that we figure if we have stuff that matches the physical nature of the organism, the more detailed that it is, it allows us to hang results on that when there's as much as possible, one-to-one correspondence between that biological nature and the code. So sorry to say a lot of things about it, but I think hopefully that covers something for you.
Can I ask, what is the sort of end game? Is it to have one model to rule them all, or is it to have an opportunity to model different things depending on what one is interested in? Can you comment on that? Yeah. I think it's definitely it's a little of both. We certainly in the project have envisioned, a family of models, some that are what I've shown you is on the end of the most complex, but less complex models to answer simpler questions are certainly, possible, certainly for speed, for optimization, and just for posing questions very quickly and iterating. But having the complex model also, right, lets us answer certain kind of questions that the simple model never could, even though it would be more computationally complex. So our end game, to some extent, at least in terms of the scientific thrust of the project, is to be able to say things about the nature of the biology of nervous system processing to a level of fidelity where the experimentalist can go back to the real worm and say, yeah, actually it is working like that. So that's why we've added so much fidelity to the nervous system in particular, is that, yeah, if there's a way for us to say that, these 50 neurons are exchanging dynamics in this pattern, and that pattern is what constitutes, say, the worm making a decision, then we want to have enough detail in our model that somebody recording from the real thing would find that pattern as well. And that's a tough thing to do, really, if you have much simpler models or much more abstract versions of the nervous system. So, yeah.
Well, thank you everyone for coming. I think it's time to end the seminar.
Thank you for all these questions, by the way. I'm a lot of folks that I haven't met in the community, but I would love to know more about each of you, and I'm sure you're all doing really exciting things. And I realize that I have been absent from past seminar events, so I will definitely endeavor to come to the ones upcoming and learn a little bit more about what you're all doing. But it's really been a pleasure to talk with you all and answer your questions. Thank you very much.
Well, thank you. We're excited to see you next seminar.
Very good.
All right.